He is a recipient of the 2022 National Goldwater Scholarship, and I'm looking forward to his PhD application for ecology graduate programs. Advyth, thank you very much for sharing your research with us. I'm looking forward to everybody's questions and looking forward to your science. You have 15 minutes. I'm going to start the timer now.
Thanks for the introduction. Hi, everyone. My name is Advyth. I'm a third-year biology major in the Spasievich Ecology Lab at UCR. And I'm going to be talking about how the mismatch between adult and seedling trees sets the stage for biodiversity change in a dry Western forest. So climate change is causing species to move, and this is really important because a key question in ecology is why are organisms where they are? The distribution of organisms is the result of a complex play of factors, and climate is one of the most important factors in determining where species are. As the climate changes, species are expected to move to remain within a climate where they can tolerate the physiological stress. And it is expected that species will move up in elevation and towards the poles in terms of latitude to remain within their optimum climate. In plants, this movement is kind of counterintuitive. Plants can't, individual plants cannot physically move the way that animals can. Instead, plants move when their seeds are transported into a new environment. And then they germinate and grow, and this is how a plant species can move. They can also shift their abundances, and we usually call this a range shift. So understanding these range shifts is really important for land management because as plant species move, so do ecosystems shift, and that affects things like carbon storage and water provisioning and other services that human societies care about. So many factors affect seedlings, and the important point here is that environmental factors can act differently on seedlings compared to adults. We need to consider this when we're thinking about how climate change might affect species distributions. Seedlings are more sensitive to heat, drought, pressure from herbivory, and are more sensitive to soil nutrients. Seeds only have a few leaves, and if an herbivore takes a bite out of them, this could be quite disastrous for an individual seedling. On the other hand, adults have had time to invest in structural adaptations that allow them to tolerate some of these stresses. Importantly, adult trees, especially in California, are very resistant to drought. Some of them can have root systems that extend into bedrock and even into aquifers. And so adult trees are much more resilient to drought than seedlings. And when we try to understand how species respond to climate change, since seedlings are more sensitive, it's important to look at how climate is affecting seedlings. So they're more sensitive to environmental conditions than adults, and the response of seedlings to environmental conditions scales up to control seedling survival. And another factor to consider is that different species have different adaptations, which results in different tolerances, and that manifests as different survival rates among species. And if you have different survival rates between species, the composition of a seedling community or seedling class can be the result of that. So for example, if oak species have higher survival rates, then they might be dominant in the seedling class, whereas if cedars have low survival rates, they might be eclipsed by the oaks in the seedling class. And the other important point is that seedlings are kind of a window into the future because today's seedlings are tomorrow's adults. If adult trees perish and there's a lack of seedlings in that area, then the species will eventually contract in its geographic distribution. Whereas if seedlings recruit into new areas, as symbolized by the light green dots, then the species distribution could actually expand into new areas that the species wasn't there before. And based on this process, seedling presence and absence is used as an imperfect proxy for future adult tree distribution. I will explain why it's imperfect later, but the important point here is that seedlings are a proxy for future distribution. And so it's a foreshadowing effect. And because seedling distribution foreshadows adult composition, we use it to study how ecosystem services might change, because adult composition is what primarily drives ecosystem services such as carbon storage and habitat for wildlife. The relative importance of environmental factors might shift with life stage, and as seen in the previous slides, this is really important in forests where there are large differences between seedlings and long-lived adults. And climate change is altering plant communities, and a mechanism by which it might affect plant communities is through controlling seedling composition. So we need to understand the relative importance of environmental factors on across life stages in order to accurately predict species responses to climate change via modeling approaches and other approaches. So I'm interested in solving this problem, and specifically I asked, do different processes control variation in adult and seedling composition? And I also asked if there's a mismatch between adult and seedling composition at my study site. My study site is the James San Jacinto Mountains Reserve in east of the UCR campus near Idlewild, and it's part of a global network of forest plots that follow similar protocols, which facilitates comparison and synthesis. My site is specifically a mixed oak conifer forest. It has an elevational range around 1,600 meters. There's 4,684 adult trees that were each tagged and measured by members of my lab previously. And there's 4 groups of trees in this plot. There's oaks, pines, and there's also manzanitas, which are actually closer to shrubs. They're fire dependent, and then cedars, firs, and others. This is a map of our forest plot where each dot represents an individual tree and each color represents a different species. Note that there's a great amount of topographic complexity in the plot. The lines represent topographic contours. So previous members of my lab measured, mapped, and tagged all of the adults in the plot, every single one. And also our lab previously measured soil nutrients and topographic variables. Last summer, I surveyed seedlings in 256 systematically arranged 1 meter squared plots. And we had 4 seedling plots nested within these larger 20 by 20 meter quadrats, which serve as a unit of replication. So in trying to understand whether different processes control variation in adult and seedling composition, we used a variation partitioning analysis that shows that the relative, it disentangles the relative influences of multiple factors in explaining variation composition. So these factors are actually groups of variables since we had so many variables. And the biological meaning of these factors is that space is indicative of dispersal processes, and environment includes variables such as soil chemistry, organic matter, as well as elevation and north and south facing exposures. And we found that the relative influences of these factors was different for adults and seedlings, which suggests that different processes might be driving adult and seedling composition. Space was more important for adults. And we also used adult composition to predict seedling composition. Maybe seedlings are driven primarily by their adult counterparts. This would make sense because, seedlings, maybe they're just very similar to the adults around them. And we found that more of the variation can be explained in using adults, but greater than 60% of the variation remain unexplained, which is interesting. So to answer that question, the answer is yes, but there's that large unexplained fraction, and that's kind of what I'm curious about for future directions of this project. The next question was, is there a mismatch between the adult and seedlings in this plot? And this figure shows, it just shows raw proportions of species. The size of the box corresponds to the relative abundance, the proportion of species, the proportion that a species represents of seedlings and adults, and each color represents a different species. And thinking back to those groups that I mentioned earlier, the oak species are greatly overrepresented in the seedling class compared to the adult class, and the pine species are greatly underrepresented in the seedling class compared to the adult class, as well as this pink shrub here, Arctostaphylos pringlei, which is also underrepresented in the seedling class. And so to test these differences statistically, we used a Permanova analysis and found that indeed the composition is significantly different. This NMDS plot on the right visualizes species composition. Each point represents the quadrat, which is the unit of replication. And if the points are farther away, the quadrats are more different in species composition. And we can see here that there's some clear clustering, specifically that the seedlings tend to be different from the adult quadrats. The figure on the right, to the right with the relative abundance is just the previous figure, but in a stacked bar plot form. We also ran an indicator species analysis to see which species are driving this pattern. And we found that 4 pine species, all of the 4 pine species in the plot, as well as one of the oak species, Quercus wislesenii, and that pink colored fire-dependent shrub, Arctostaphylos pringlei, are driving the trends that we observed. So the answer for this question, is there a mismatch, is yes, but we actually don't know what's driving this mismatch. My data only identifies patterns. It doesn't necessarily directly measure processes. So in terms of the different processes controlling variation in question 1, an important implication of this work is that modeling approaches might need to consider that the relative importance of these processes in structuring communities can shift with life stage. And this doesn't necessarily just apply to seedlings versus adults. This trend could potentially be generalized to other life stages, but of course, more work is needed to be certain. And in order to address the high degree of variation that was unexplained, in order to improve the predictive power of our statistical modeling, we will install temperature sensors because microclimate might be playing an important role. A lot of literature suggests that understory temperatures are greatly different than ambient air temperatures above the canopy. And we think that microclimatic variation may not be captured by the variables that we have. And so in order to quantify microclimatic variation, we're going to install temperature sensors this summer. In terms of the mismatch, it suggests that there's a potential for change in this forest, but we can't say for certain if a change is going to occur. We have observed very low pine recruitment and extremely low oak recruitment. That is a lot of oak seedlings and very few pine seedlings. And we've also observed large-scale pine mortality in and around the forest plot. In this picture, each arrow is pointing to a dead pine tree canopy. And you can see that there's a significant amount around the forest plot. And our results suggest that there is a potential for a shift from an oak, mixed oak conifer forest to an oak-dominated forest. And if this were the case, it would be consistent with regional trends that have been observed in California, as well as global trends that have been observed around the world. The figure on the right shows, is from this 2018 paper that did a literature review and found that a great majority of studies around the world have documented shifts from pine to oak species in forests. And so the red and orange represents a pine to oak transition. This trend is also consistent with patterns that we observe in California's paleohistorical record, where during warm interglacial periods, oaks became dominant over pines, whereas in cool periods, pines were dominant over oaks. And so if these patterns, if modern contemporary climate change were to cause patterns, we expect that it would mirror patterns that were observed in the past as well, in the distant past. However, again, we can't say definitively whether a shift in forest composition will occur. We need data over much longer timescales, especially since many species recruit periodically, and maybe the year I sampled was just a bad year for certain species. And so we plan to resurvey the seedling plots. We also want mortality rates for each species to assess whether whether some species have low mortality rates or higher mortality rates, and understand whether these mortality rates are changing. We also can't attribute these patterns to climate change with the data we have necessarily, because fire suppression is a confounding factor. On the bottom right, we have Smokey Bear. We don't like Smokey Bear very much here in Southern California. Smokey has caused our forests to be much denser than the natural level. And this actually causes the forest to become a lot shadier than they would otherwise be, which actually inhibits the growth of some of the more sun-loving species like the pines, and promotes the growth of shade-tolerant species such as oaks. And so the pine oak trend could also be driven by fire suppression, or it could be driven by climate change, but they're not mutually exclusive. So overall, we need long-term data to assess whether a change is occurring. And in order to do this, we will resurvey the seedling plots this summer, and then resurvey the plots every year thereafter. And we're also interested in installing seed traps to see whether seed production might be driving the patterns in seedling composition. Maybe some species just aren't producing enough seeds. And so that's another factor we want to disentangle. So as a broad summary of what we discussed, some processes were more important for seedlings than for adults, and this affects seedlings composition. In the composition, we found that there was a significant mismatch between adult and seedling trees at the site. And in terms of the implications for species distributions, we actually need more data to know if shifts are likely to occur. And we will get that data via resurveys and other long-term monitoring projects at the forest plot. And if we can understand how these factors play out, we will be able to improve our predictions of species ranges and abundances, which will help us conserve important ecosystem services in order for human society to flourish under this period of contemporary warming. I would like to thank everyone who has provided mentoring in this project, as well as members of my lab who collected some of the data that I used in my analysis. And I want to thank the Natural Reserve System and the Native Plant Society for providing me funding to work on this project. Thank you.
Advyth, that was very, very good. Thank you very much for your presentation. Do we have any questions? Rhea has one in the chat. Rhea, are you able to unmute and ask Advyth?
Yeah. So you mentioned that you plan on getting data on the mortality rates of the different plant species. Do you have an idea of how you would calculate this?
That's actually a really good question. I've been thinking a lot about that recently. There's simple ways to calculate it, like just how many, what percentage of seedlings progress from the seedling class to the sapling class. So you can look at rates of transition. You can also look at like what percentage of seedlings die per year. So you can look at rates over time. We can also look at rates over spatial variation, so like percentage of mortality per quadrat. But I definitely, this is on the to-do list in terms of understanding what kind of like modeling approaches might be appropriate to understand mortality rates. But yeah, that's a really good question. There's a couple ways to measure it.
These long-term projects are very suited to a PhD. Not many people know this, but my degree was actually in ecology. So I remember those quadrant surveys very well, except I had a lot more sphagnum and a lot less rattlesnakes than you did from the sounds of it. In Scotland, we don't get many of those. So I was going to ask you about this idea, the amount of biology that you have to incorporate into some of these questions, and how much is known about seed production by your different species. And is there a way to, I love the Idlewild area, and I've seen those awful dead trees too. Is there a way to incorporate a different site to act almost as a control when you do those seedling surveys?
Yeah, so I know that Well, my PI, Marco, who is here, has
Oh,
planned to
good
have
job.
satellite plots to compare what's going on in the main forest plot to. I had planned a transplant experiment to look at not seed production, but specifically survival rates across different elevations, but I wasn't able to. I'm not able to do that this summer because turns out nobody in the state of California is growing the species that I need to plant. And so that's one of the things we might do is actually just grow our own
You
seedlings.
need some of those seeds.
Yeah, so we'll have to look at that as well.
Yeah, it's a really interesting project. Do we have any other questions? Okay, thank you very much, Advyth, for your great work and